You've probably already studied how the muscles work. Now we need to see the relationship between the brain, the central nervous system, and the messages that are sent to the muscles in order to contract. Today, we're going to explore the neuromuscular system. Hi everyone, welcome back to the Physical Educator. Today we're going to talk about topic 4, movement analysis. It's a short unit with only 4 topic points, so let's look at them straight away. It's a short unit with regards to the specification. However, there's 3 areas where you need to go into a bit of detail, particularly the sliding filament theory, arguably the hardest part of the course. With that said, 3 out of the 4 explained, so we need to put a lot of meat on the bones here. So without further ado, let's get straight into it. The first 2 topic points are looking at labelling the motor neuron and also looking at the neurotransmitters that enable the impulse to pass across the synapse. Before we look at labelling the diagram, let's have some understanding on what we're looking at here. And we're looking at the brain, the central nervous system, the peripheral nervous system, meaning the motor neurons, connecting with the muscles to send messages from brain to muscle in order to contract. That's what we're looking at here. And the motor neuron is the deliverer of that message. So the motor neuron itself, how it looks and how we need to label it, we start off with the dendrites. They're the tentacles that receive the information that's being sent down the central nervous system. Inside the dendrites, we have a cell body, like with every cell within our body. Inside the cell body, we also have a nucleus. Again, no surprises there. We know that with cells, we know they have a nucleus. It's where the cell's activities are coordinated. This is where this separates from other cells that you've probably looked at before. We have a thing called an axon. Now, the axon is just a long transportation device down the middle of the neuron. So it's received at the dendrites, it's calculated in the nucleus, and the impulse is then sent down the axon. Now, the axon can be 2 centimeters up to over 1 meter, depends on where this neuron is in the body. But the impulse needs to be sent down it nonetheless. How it travels down, complicated method, it's called an action potential, and we're going to keep it nice and simple and just say that sodium ions and potassium ions interact with each other to transport this impulse down the axon. But it's still an electrical action potential, meaning there's potential for action, but it's not happened yet. Now, when it reaches the end of the axon, it reaches the axon terminal. Just like when you go to the airport, you're at a terminal. It's the end point before you transport somewhere else in a plane. This is the same for the impulse. It's the end of the axon before it branches off right, left into the synaptic end bulbs, which there's many, and they are attached to a muscle. But we'll look at that attachment in a second because it's not as obvious as you would think. Now, with the synaptic end bulbs, they are what's part of the neuromuscular junction. This involves the synaptic end bulb, the muscle itself, and as you can see in this diagram, the little receptors that sit just on the muscle tissue. Now, what's important for us to know here is that this action potential has travelled down the axon. It is now depolarized the synaptic end bulb. What that means is, in layman's terms, calcium ion gates have opened, calcium ions have rushed into that synaptic end bulb, and they've pushed these sacs that you can see containing acetylcholine towards the surface, towards the end of the synaptic end bulb. And as you can see from the bottom right one, it has merged into the membrane, and acetylcholine rushes into the synapse, and it finds the receptors to sit on of the muscle. Now, these receptors that it sits on are vital because they allow the sodium ions into the muscle cell, and this transports the impulse from the neuron into the muscle. So the calcium ions allow the acetylcholine to enter the synapse. They attach to the receptors, and sodium ions can rush into the muscle cell. So we've just spoke of acetylcholine, and that is the neurotransmitter that allows the impulse to pass from the neuron into the muscle. But when this needs to stop, cholinesterase is released. It's a catabolic reaction. It breaks down acetylcholine and it disperses it. Therefore, the receptors shut. Therefore, no impulse can pass across into the muscle. therefore there's no more muscle contraction. And that's how our body manages the ability to send a message from brain to muscle. And we have a contraction, and then the contraction can stop. That's all we need to know here. We need to label the diagram. We need to know about acetylcholine and cholinesterase, and a little bit of understanding about how the impulse is sent from the brain to the end of the neuron into the muscle cell will also help you with your knowledge for the sliding filament theory. Depolarization is a key word here to remember. It's definitely a mark that you can gain on the sliding filament theory by saying that the synaptic end bulb depolarizes, the neuromuscular junction depolarizes, and this allows the acetylcholine to sit on the receptors to allow sodium ions into the muscle cell, which triggers the start of the sliding filament theory. Next up, arguably the hardest aspect of the course, we've got to be able to explain how skeletal muscle contracts by the sliding filament theory. The sliding filament theory is a difficult concept, so we're going to think about it in a different way. We're going to think about it as a prison break, releasing hundreds of inmates. But firstly, we've got to get into the prison, get to each cell, open each cell door at once, and release all of these inmates. And our 5 defendants are here in the prison cell. They're responsible for different aspects of this prison break or sliding filament theory, but what they are responsible for is unknown at the moment. So we've got sodium ions, potassium ions, calcium ions, acetylcholine, and cholinesterase. Like I said, each of them responsible for something, some responsible for more than others. What we need to do before we look at their crimes is understand the 5 crime scenes. Now what I've done here is we're going deeper and deeper into the muscle with each crime scene. So Exhibit A, depolarization of the motor end plate. This is what we've spoke about already, where the acetylcholine is released from the synaptic end bulb across the synapse, sits onto the receptors, the sodium receptors on the muscle cell, allowing a rush of sodium ions into the muscle to allow the muscle action potential to go further into the muscle to the sarcomeres. Cholinesterase covers the tracks of the acetylcholine so it's undetectable, so we don't even know that it's involved in this crime. It breaks down the acetylcholine and that no more contraction can occur from that moment on. We've spoken about that already, so let's move on to Exhibit B. Let's go a little bit deeper within the muscle. Exhibit B is the sarcoplasmic reticulum. Now, this sits around the muscle fibre. Remember, the muscle fibre is attached to the neuron, so every single filament, every single myofibril within that muscle fibre is stimulated by this impulse. And the way it's stimulated by this impulse is how it's transported around the muscle fibre. And the sarcoplasmic reticulum sits around the outside of the muscle fibre, so the impulse can spread to every myofibril within that muscle fibre. But we also have the T-tubules, and these go on the lengthways of each myofibril, so even every sarcomere can be impacted by the sodium ions released into the muscle fibre. Going further than that, Exhibit C, we have each sarcomere. So what you can see there is a little cross-section of a myofibril. And each sarcomere is separated. And this is the reason why a muscle is striated in colour, because you can see the Z lines and you can see each of the ends of the sarcomeres. But we need to go even deeper than that. And Exhibit D is the myofilaments within the myofibril, within the sarcomere, which is the actin and the myosin. 2 very important things to remember here and 2 easy marks to pick up. The H zone, the section in the middle on the top picture, when the muscle contracts, the H zone disappears as the actin and myosin pull over each other. The Z lines or the Z discs on the side of each sarcomere, they also shorten as each sarcomere shortens, and when each sarcomere in the whole muscle fibre shortens, the whole muscle shortens, and therefore we have concentric contraction, and that's pretty much what we're talking about here. But we need to know a little bit more information if it's a 6 or a 7 mark question to enable you to get every mark. So we also need to consider the troponin and the tropomyosin. And this is where Exhibit E comes in. These are the bodyguards, and we have to move these bodyguards out of the way in order for the actin and myosin to access each other. So before we move on, they're the 5 crime scenes. I want you to revisit this bit over and over again. Make sure you're familiar with the location of what's happening within the slide and filament theory, and then we can explain the chain of events from there. Okay, so you should be ready to explore the chain of events now. We've looked at the 5 guilty personnel, we've looked at the 5 crime scenes, we're now going to look at the chain of events and what happens. We've already spoken about this a number of times. First of all is the depolarization of the motor end plate. We need to get this action potential from the neuron into the muscle in order for the muscle to be stimulated. And acetylcholine is released, assisted by calcium ions, which is triggered by the influx of sodium ions into the motor end plate, which depolarizes it and allows this acetylcholine to pass into the synapse, sit on the receptors, allowing sodium into the muscle. We now are in the muscle cell, and we have a muscle action potential, and it's an all or nothing action potential. The whole of that muscle fiber will either be stimulated or not stimulated at all, and that's what we call the all or nothing muscle action potential. Here, it travels around the sarcoplasmic reticulum, and the sodium passes down the T-tubules until it arrives at each and every sarcomere. Again, we've explained that already. What happens next is similar to when the motor end plate gets depolarized, a similar concept here, but at every sarcomere. And this is where we get rid of the bodyguards, pretend the prison break, pretend that a bodyguard is outside the cell block and you've got all of the cells inside the prison. What we do is we allow the calcium ions in there. The calcium ions bind with the troponin, which is connected to the tropomyosin. So when a protein binds with something, it changes its shape. So the calcium ions bind with the troponin, change shape of the troponin and tropomyosin, and this allows them to move out of the way, exposing the active sites of the actin for the myosin to grab hold of them. It's almost like taking the bodyguards out of the cell block, unlocking the cell gates, and allowing all of the cellmates to escape. Once this has happened, and the actin and myosin can form together to create what's known as a cross bridge. And this is where the myosin grabs hold of the actin and it's ready to initiate a power stroke. Now the power stroke comes when the ATP releases the energy, obviously turns into ADP plus P, but the energy is released and this allows the slide to happen. Very small slide. Once this has happened, the myosin lets go of the actin, and as long as ATP is present, it can continue to do this again. As you can see, the power stroke pulls the actin filament past the myosin, shortening the sarcomere between the Z lines. The H zone disappears. This happens in every single sarcomere within the muscle fiber, and that's when we have a concentric contraction. Like I've just said, this continues as long as there is enough calcium ions and ATP present within the sarcomere for this to take place. And it will continue and it will continue and it will continue. If this was a prison, we'd have riots outside because there's so many inmates that are escaping, but we're not in a prison, we are in a muscle, and if it continues to happen, we get muscular contraction until further notice, till we don't need it anymore. One final recap: sodium and potassium interact down the axon, depolarizing the motor end plate. Calcium ions rush into the motor end plate, forcing acetylcholine to blend through the membrane into the synapse, sit on the receptors, open up the sodium ion gates, allowing sodium ions into the muscle. Cholinesterase breaks down the acetylcholine and no more sodium ions can pass into the muscle. This causes a muscle action potential. Sodium ions spread throughout the whole muscle fiber until they get to the sarcomeres where they release calcium ions once more. Calcium ions allow the change of shape to the troponin, which in turn changes the tropomyosin as well. This reveals the active site of the actin. As long as calcium ions and ATP is present, we can keep creating the cross bridge, we can keep creating the power strokes, and we can keep shortening the sarcomere. This continues until the calcium ions or ATP are not present, and then there's no more muscular contraction and we can stop. A very difficult concept. I've tried to split this up into sections for you, so hopefully you can take in all the information. My advice with answering a question here is look how many marks the question is worth. If it's worth 6 marks, then you're going to need quite a bit of detail to get your 6 marks for the sliding filament theory. If it's only after 2 or 3 marks, then it's going to be talking about the calcium ions, moving the troponin and tropomyosin, exposing the active site of the actin, allowing the cross bridge, allowing the power stroke, and the shortening of the muscle happens. So it depends on how many marks the question's asking for what you need. But for your own knowledge and your own understanding, it makes sense to do a bullet point list of the concept of the sliding filament theory, maybe an 8-point list to make sense of it, and you can take that with your revision ready for your exam. Like I've said, it's a difficult concept, one that needs a bit of revision, needs a bit of repetition, repeat until you understand the process. And there's no irony involved there, because obviously it repeats as a process itself. And finally, we're going to look at slow and fast twitch fibres and how they differ in structure and function. So slow and fast twitch fibres, we're talking about recruitment here. And what this means is the neuron you can see on the right hand side is attached to each of them muscle fibres. So the sliding filament theory will occur in each 4 of them muscle fibres if an action potential was initiated. The bigger the recruitment, the more forceful the outcome. The smaller the recruitment, less force, but because it's a smaller recruitment, it has a little bit more resistance to fatigue, so it can go a longer duration. Already you're probably developing an understanding of the difference between slow and fast twitch fibres. Let's look at some of these sports. If you were to consider the energy systems, looking at some of these sports, you would be able to say some of them require the aerobic system, some of them are an explosive movement, therefore it might be one of the anaerobic systems, etc. And your muscle fibre types, you can consider in a similar manner to the energy systems. But no matter what sport it is that you're playing, a muscular contraction is required. So what does it mean, twitch fibres? Well, you've got slow twitch fibres, and literally it's a twitch, so it sounds like this. Or you've got fast twitch fibres, which sound like this. As you can sound already, there's a difference. The slow twitch fibres are slower, more steady contraction. It's not going to generate all the power, but it's going to last for longer. And they're called oxidative fibres because oxygen is present within that muscular contraction. They're also called type 1 fibres. Then we've got the fast twitch fibres, and we've got 2 different types. We've got the type 2B fibres, which are your explosive fibres, no oxygen present, big, powerful, short contraction. Then we have the type 2A fibres, which is like a love child of a type 1 and a type 2B fibre. Let's explore this a little bit further. Slow twitch fibres. The structure, size of the motor unit, so that's back to that first picture we looked at. That means that the motor unit is how many muscle fibres are attached to the neuron. It's a small motor unit, meaning there's not too many fibres attached to the slow twitch fibres. Mitochondrial density is very, very large. Capillary density is very, very large. The oxidative capacity is very, very high. This is because oxygen is required. ATP takes place in the mitochondria for aerobic respiration, so therefore the density is high. Capillary density, well, if we have more capillaries, we have more oxygen passing through into the muscle by gaseous exchange. Therefore, it must have a high oxidative capacity. It goes without saying. Glycolytic capacity, so its capacity to use glycogen as energy is very low. The function of a slow twitch fibre, its resistance to fatigue is very high because of the oxidative capacity that it has. The force production is very low because of the size of the motor unit, and it recruits less muscle fibres. Therefore, it's more of a jog compared to a sprint. You imagine that yourself. When you jog and when you sprint, how much more of your quadricep muscle and your leg muscles do you feel like you're using when you sprint? That's because more muscle fibres are being recruited. Activities used for distance events, duration sports, and your fuel source within slow twitch fibres is your triglycerides. Going into type 2B fibres, the complete other end of the spectrum. Size of the motor unit is very, very large. It's a forceful contraction. You need all the help you can get from your muscles. Therefore, it recruits as many as it can do within that muscle. The mitochondrial density is low. There's no oxygen required, so the capillary density is low. The oxidative capacity is very low. once again because oxygen is not required. Glycolytic capacity is high. Glycogen and glucose is our main fuel source here. So therefore, the capacity to use that for energy is high. The function, resistance to fatigue, low, obviously. Low mitochondrial density, low capillary density, low oxidative capacity, that goes without saying. Force production, very high, because the motor unit is very large. Therefore, we generate a forceful contraction, forceful movement. Activities used, short sprints, explosive actions within games. A footballer or a basketball player, they still have explosive actions even though they require a lot of oxygen and a lot of aerobic capacity to their sport. They also require explosive actions within their sport, and that's where the type 2B fibres can be used. Fuel source, CP and glycogen. And finally, we have type 2A fibres. Now, type 2A fibres are the love child of a type 1 and a type 2B. So just take all of the best qualities from both fibres, but not quite as much. So the size of the motor unit is large. It's not very large like the 2B, but it's large. So therefore, you can still create quite a forceful contraction. Mitochondrial density is also large. Again, not as large as the type 1 fibre, but it's still large, meaning it has some oxidative capacity. Capillary density is large. Again, oxygen is required, hence a high oxidative capacity, but also a high glycolytic capacity because glycogen is used as the source. This fibre type can be referred to as the oxidative glycolytic fibre type because both are required. The function, resistance to fatigue, yeah, it's got a bit. You know, lactic acid will build up, but it's got some resistance to fatigue, more so than the type 2B fibres. It can produce a high force outcome, not quite the same as a type 2B fibre, but it's still a good forceful contraction that can occur. Activities used for 400 meters, 800 meters, prolonged anaerobic bursts that last longer than 15-20 seconds. Obviously, you can't go 100% for that long, hence the reason why the forceful contraction is slightly lower than a type 2B fibre, but you can still create a strong output for a longer period of time using these fibre types. Fuel source, fats and glycogen. So when you look at this as a summary across all three of them, what I want you to look at is the comparison between the type 2A fibres and the type 1 and the type 2B. And the way you do this is by looking at the type 1 and the type 2B fibres first, because they're completely different, the other ends of the spectrum. And the 2A fibre just takes the best qualities from the type 1 and the type 2B fibres to make this super fibre type, if you like. It can't beat a type 1 fibre in its own right, it couldn't beat a type 2B fibre in its own right, but it's a fibre type that takes both qualities for them sports that require forceful contractions, but over a longer time than a 5 or a 10-second period. One thing to remember with fibre types, an athlete doesn't just have one type of fibre type. So Mo Farah doesn't just have type 1 fibre types. There's going to be certain muscles within his body that are dominated by type 1 fibres, but he also has type 2A and type 2B fibres. Let's look at Usain Bolt, the same, dominated by type 2B fibres, but it's not the only fibre types within his body. He also has type 1 and type 2A, just to a lesser degree. So that's really important to remember when you're looking at fibre types. It isn't that an athlete just has one type. They do have all three, but the percentage of the makeup of them, not only in the whole body, but in certain muscles, is different. And that's what makes them different athletes to other people. Don't forget to subscribe to the channel. If you're looking for teaching resources, check out the TES website and go to the Physical Educator shop. You can pick up the whole unit, movement analysis, looking at all aspects, including a prison break challenge for the sliding filament theory, joint movement analysis, and a full comprehensive biomechanics section 2. We also consider IA suggestions as well as practical lab ideas. Check out the resource in there. Good luck. See you again soon.